ONC Live Insights is a video editorial program produced by ONC Live.
One class of drugs that has had some level of interest in the ALK population, other than the tyrosine kinase inhibitors, has been the HSP90 inhibitors. All proteins, for their conformation to be effective, require kind of a scaffolding effect, and that's what HSP90 is. So when you inhibit the scaffold that holds together the protein in its functional capacity, the protein becomes non-functional. Now, all of these oncogenic drivers require HSP90 for their function. But one of the ones that seems to be most sensitive is the ALK oncoprotein. And so some of the initial trials with HSP90 inhibitors noted quite a high response rate with several HSP90 inhibitors, and it was pretty consistent across the data with gantenerumab, data with AUI-922 and other HSP90 inhibitors, that in the ALK population you saw objective responses that were actually quite notable. Now, the HSP90 inhibitors are a class of drugs that have not yet been successful in an FDA approval strategy to date. I think the interest in these agents somewhat dissipated with the increasing numbers of tyrosine kinase inhibitors, the alectinibs, brigatinibs, ceritinibs, those sorts of drugs. There is some data combining HSP90 inhibitors with ALK inhibitors. That data is very early. We don't have a good read on it yet, but that's one strategy where perhaps the combination of an ALK inhibitor, perhaps in refractory disease, would be a viable strategy. I would also think that if I had an ALK patient that was refractory to a series of tyrosine kinase inhibitors, I might look for a clinical trial with an HSP inhibitor to enroll that patient on. So that's something that I would think about in an ALK-positive patient. The age of immunotherapy is upon us. We have FDA-approved options for patients right now. One of the interesting observations about immunotherapy is that there seems to be a link between the effectiveness of immunotherapy and the mutational load we see in the cancer. If you look at mutational load in all cancers, the two most highest ones are melanoma and lung cancer. That's where immunotherapy has been successful. So there's a suggestion that the more mutations you have, the more neoantigens you may have, the more immunogenic your tumor may be, and the more successful immunotherapy may be. Now, let's look at the other end of the spectrum. It's very clear that never smokers, which comprise the population of EGFR mutants, ALK-positive patients, those sorts of things, they have a much lower mutational load. And I must admit, the early data, not so much in ALK-positive patients, but in EGFR mutation-positive patients, when you look at the differential benefit from immunotherapy versus standard chemotherapy, there really isn't an incremental benefit, suggesting that immunotherapeutic agents may not be any more effective than standard chemo. They may be better tolerated, but they aren't more effective. Whereas if you look at patients who have a smoking history or a higher mutational load, there's a clear advantage of the immunotherapeutic agents over standard chemotherapy. So that brings us to what role is immunotherapy going to play in the oncogenic driver subset, such as EGFR mutations, ALK translocations, ROS1. How should we combine them with the available TKIs? I think these are important research questions, but I think many of us have a much more cautious level of optimism that the immunotherapeutic agents are going to be a home run for these population of patients. Although, the data is very early, the data is not completely compelling that immunotherapy is going to be a home run for these patients, but it may be that during the life cycle of the tumor, that changes within the tumor allow it to be more immunogenic, and immunotherapy may be an option in combination, or either by itself or in combination with TKIs. There's actually been very little data presented to date on the combinations in the oncogenic driver subsets of patients. So we have to draw our initial impressions from the phase 3 data, such as the Checkmate trials that in Checkmate 057, the never smokers and EGFR mutants really didn't have a benefit relative to docetaxel. So I think many of us are a little cautious about what role these agents may play, either as monotherapy or in combination in the oncogenic driver subsets.
There's a lot of excitement about potentially combinations of ALK inhibitors with immunotherapies, with the hopes that we may be able to induce even longer remissions for patients by incorporating an immunotherapy into the regimen. I think it's too early to know. These trials have just started. These are all phase 1 trials, and there are a number of them. Ceritinib and nivolumab are being combined in a phase 1 study. Alectinib and atezolizumab are also being combined in a phase 1 study. And the newest ALK inhibitor, lorlatinib, is also being combined with a PD-L1 inhibitor. So a number of approaches to trying to combine TKI ALK inhibitors, TKIs, with immunotherapies. We don't yet know how active these combinations will be, if they really will make a difference, but I think there is a potential concern for toxicity with these combinations. We've already heard about potential increased pneumonitis with the combination of EGFR inhibitors with checkpoint inhibitors, and so with these phase 1 trials now launching of ALK inhibitors and immunotherapies, we're really going to be on the lookout for any increased or unexpected toxicities.